Right, so that was Friday, end of week 3 of teaching at the University of Bristol. So there's a day out of Bristol today. The only scheduled thing today was some synchronous teaching. It was a practical. It was a 2-hour session of stations, essentially, in which we went to the veterinary dissection room that's on Southwell Street, and we went to the labs there, and there were 5 stations set up. The 5 stations included, one was looking at ultrasounds, another at radiographs, a third on equine terminology. So this is essentially the lingo that horse people would use. So stifle joint, withers, pastern, things like that, really. But like essentially the non-scientific terminology that horse people use. So it was labelling that, and then another was about bone structures generally. So one was talking through essentially, well, we got given the option to either. So there's this room essentially in the building that has like tons and tons of like skeletons and specimens that you can freely go into and use as a study space. And they essentially gave us the option, like, do you want to like walk around freely, are you familiar, or do you want to talk us through it? And we essentially reconciled that to talk through it. And they essentially walked us through the skeleton bone by bone. I pretty much showed off and called out most of the names, but that was only because no one else did. I tried to give people the chance to respond sometimes. Some things I didn't know, so there was the like the pelvic bone, for instance. I forget the name. I remember one of them, but the ilium, one part of the bone, but then I forgot the other 2 parts. I managed to familiarise myself with the bones, because I think that's an important part. You know, it's talking about anatomy. You know, I think it helps to know the bones, the bone names, and identifying them. So, like with the vertebra, for instance, you got like the spinal process is quite pronounced on the thoracic lumbar, and less so on the thoracic vertebrae, and less so on the lumbar vertebrae, in which you have the transverse process, in which would be the what goes outwards. And essentially also like the reasoning for this is because of that essentially holds the mass of the abdominal cavity up, because you don't have essentially the ribs there, it's just a floating mass. So you need that like extra transverse process that is essentially like the outward bits, because that helps with that. So it's all quite interesting. So there is a sort of like an anatomical reasoning for the structure of the bones. So that was actually quite interesting. It was quite interesting to go through, talk about some things, ask some questions, talk a bit about the calcification, the keratinization, or the ossification and the keratinization of bones. So that was quite interesting, very, very interesting. It was fun to get like a nice practical thing, because it's one thing, because you could go through on a presentation, a PowerPoint, and you could go like, oh, what's this bone and what's this bone? They just show you an image, and then you say, oh, what's this bone and what's this bone? And like, whilst you can use that as a means of learning, it is good to get that visual aid. That visual aid is nice, and it's just more fun as well. It's more fun to like interact and like call this stuff out. So that was good. And then the final station was referring to I believe it's the topographical terminology, so talking about dorsal, ventral, cranial, rostral, talking about the different forms of locomotion, so flexion, extension, supination, pronation, those sorts of things. So that was quite good. And then there was a specimen of a dog that had been split in half on its medium plane. So, and then you got to see the inside half, and then you got to see it from an outside half. So that was quite cool, and essentially probing the lecturer on, because of course I've been a butcher in the past, I was curious as to how, like, oh, is that an actual job? It's someone's job who actually prepares these specimens. And then talking about, say, with beef, for instance, if you want to say like a full quarter of beef, or if you wanted to say a full cow, that's going to cost you, that's into the thousands of pounds for buying beef, and then it's, that costs more because that just goes for meat. So I was asking them, so when you get onto the dissection stages and looking at anatomy and so forth, what sort of specimens would you be coming across? So dogs, so like the dogs and cats essentially would be donated and they're free, so you'd be able to get access to more or less everything. But when looking at, say, like organs, for instance, looking at organs, you would be looking at you could be looking at bovine and the pig, and looking at the offal essentially that doesn't get used by the meat industry, essentially, because it essentially has no value. So like the small intestine has no value in entering food production, essentially. It doesn't get used, whereas obviously all the meat and the bones and stuff, all that enters the food industry process, so it has value. So essentially the stuff that in the abattoir that essentially wouldn't enter the meat process, enter the food industry process, would be the stuff that goes to the labs for research and for students to work on. So that essentially makes sense. But it was curious to see, like, it's actually someone's job to dissect these dogs, and then like they go through like a preserving process. So they use a formaldehyde and they put it in water to preserve it, and like they're years and years and years old. They're like some of them are very old specimens. So there is that slight discoloration because they're not bright pink. But obviously, when it comes to getting the, say, like the bovine organs, for instance, that stuff can be a lot more fresh, which I think would be quite nice, because it's going to be quite interesting there. Because when it comes to surgery and operations, you wouldn't operate essentially on cattle and pigs just because there's no money in it. You'd essentially you would just euthanize the animals, and they would go for meat, because farmers don't have the money to do these massive ops on their animals when they would typically just be in the meat process. People don't really keep pigs other than to grow them up for meat. It's just not a thing, lambs, beef, pigs. This is all livestock. It's all got essentially a lifespan, which it all enters the system for human consumption. There are people who do keep these animals as pets, but that is a very rare number. So, but anyway, yeah, it was all very interesting. A very interesting session. We then went to a quiet place. We went to the chemistry building and just worked on some previous work. We did a physiology catch-up, or physiology asynchronous task, which was about osmosis and tonicity, which was wasn't interesting, but you do need to know it. So how fluid and solutes move within the body, and like the terminology you would use for that. So like the osmolarity and the molarity and the tonicity, and talking about hypertonicity, hypotonicity, isotonicity, and then talking about a process in which the tonicity inside cells is regulated through a sodium-potassium ATP process to prevent hypertonicity, and then the impacts of hypertonicity in which you get cell bursting. So yeah, rather interesting stuff and need to know stuff, but it wasn't particularly interesting. We then did the equine stuff, which is very basic. It was talking about basic equine care, basics of what equine and horses are used for, the management processes, the breeds, just getting a quick jot note take of that, so that didn't take very long. I don't have much interest in horses and equine, but it is a big part, and you do need to know it. And then lastly we did, well, it was the first thing we did, but it was on cell growth, cell growth and development. So talking about muscle cell growth, talking about the like the cell growth processes. So like your pituitary gland is responsible for producing the growth hormone that produces this. I think it's a hormone called mTORC1. And that's the major cell growth factor. And talking about how there's inhibitors that can affect this, and there's mutations of genes that can affect this, and then that will determine cell growth. And they're talking about how nutrition and exercise can inhibit cell growth as well, and how that stimulates this mTORC hormone, in which you will get that essentially cell growth. So yeah, that wasn't too bad, but we got that down. So yeah, so some interesting stuff. I did get a bit more than I was planning on doing today. We also had a quick meeting with our personal tutor, which was again very basic. It was just a meet and greet, more or less, an introduction, and then that whole process with the personal tutor is going to be on monitoring our progress. And then it's also a means by which we can address any major concerns if we deem we need to with them. So yeah, so we had that as well, which only took about 20 minutes, so that was all right. And then we got a meet up in a couple of weeks' time with them, so that's okay. So that's it for week 3 of teaching. Next week, again, it's another week of teaching, obviously. We got another animal handling practical going on. We got continuing with our CBL stuff on Monday, the puppies and kittens CBL task. And yeah, proceeding henceforth. I have 2 cell biology async tasks to do. I have 2 agents of disease async tasks to do, and then we have some CBL work which was due for Monday. So I'm thinking the plan for the weekend is, I'm interested in the agents of disease, so I'll probably do those 2 tasks this weekend, do the CBL task for Monday, and then we'll have the cell biology on backbench for next week to do in breaks, because it's good I think it'll be good to have that when I get a break, because there will be points where I'll have like, I'll have an hour break, or I have a couple hour break here. And what I don't want to basically sit on my ass and do nothing. I mean, whilst we could look at old previous notes and jog our memory, I think it is quite handy to like not force ourselves to always pressure ourselves on that work. It gives me that, okay, well, we can catch up on this. But obviously we don't want to have 10 things to have to catch up on. We want to just like keep that backlog of work as low as possible, but it's not the worst thing to, if we do have a couple things outstanding, it's not the end of the world. But as long as yeah, we don't have, so like today for instance, we managed to knock 3 off, and we'll be able to knock a bit more off on the weekend now, so that's good that that's all knocked off. But hypothetically, say we didn't do what we'd done today and we didn't do anything on this weekend, like we have a bit of a backlog as more stuff will pile up next week. So yeah, just keeping on top of it, keeping yeah, just keep on jogging our memories as best we can, try and remember a lot of these things, because some things I have been like the bone and stuff and anatomy and the topographical stuff, like that stuff I've been regularly jogging my memory on. But some things, such as like cell biology, for instance, or perhaps things like osmosis, there are like elements, I guess, that's like, oh yeah, we actually completely forgot about that, or oh yeah, we did learn that, but it has completely swept our mind, and we do actually need to incorporate it. But again, it's all still implementation. We have implemented some of it more than others, you know, we have implemented more than others probably have, which is good. So like there, at least we've taken some stuff on board, and it has actually like quite fixed nicely into my memory, which is good. So like the bone stuff, like that's quite solidly fixed now in my mind, which is good. And I also have that visual aid, because I think the visual aid is always nice, because with something like, say, cell biology, and you're talking about like specific hormones or specific terminology, like I always find that like a good physical aid is always helpful, and especially like if there's like a physical event associated with it, I can usually remember it a lot better. Because if you've just read about it and I've written it down, like the writing it down helps. But if I have if there is essentially a physical task that I have previously done that will help with it, then I think that's quite good. So with the hoof, for instance, the hoof of the horse, I learned that prior to today's practical, so I did have a vague understanding going into it, but it wasn't cemented into my mind. But today, for instance, we had some hooves in which we could like stick labels to, and like now that's in a fixed memory, so I can be like, oh yeah, that was that practical that we'd done in which we did such and such with such and such, and like we were in this environment, and like I remember putting on the labels and stuff, and like, oh yeah, I actually had that visual aid. So I thought that's quite a good thing, having that visual aid. At least it helps me, at least. But anyway, we'll stop there. Yeah, next week again, yeah, just carry on. We'll get some more work done this weekend, get some CBL stuff done this weekend, have a tiny bit of outstanding for next week, unless we get all of it done, the cell biology and the agents of disease stuff done. We'll see how we go. We certainly could get it all done this weekend, but we'll see how we go. The parents are back from Scotland tomorrow, so yeah, it'd be nice to see them and see the pooch, because the pooch is back now, the doggo. So yeah, nice to see him again, just because it's a bit quiet in the house at the moment and a bit unproductive, a bit much quieter, but yeah, we'll crack on and we'll catch in with you next week.